Part 1. Glycolysis. Glycolysis is the first step of carbohydrate metabolism, where glucose is broken down into two molecules of pyruvate. Glycolysis occurs in the cytoplasm of the cells, and does not require oxygen. It can be used by both aerobic and anaerobic organisms. Glycolysis consists of 10 reactions, each catalyzed by a specific enzyme. The reactions can be divided into two phases, the preparatory phase and the payoff phase. In the preparatory phase, two molecules of ADP are used to activate glucose and split it into two molecules of glyceraldehyde 3-phosphate, G3P. In the payoff phase, each G3P molecule is oxidized and phosphorylated, producing two molecules of NADH and four molecules of ADP. The final product of glycolysis is two molecules of pyruvate, which can enter different pathways depending on the availability of oxygen. In this part of the video, you will see the animation of the glycolysis pathway, showing the structure of each molecule, the name and function of each enzyme, and the energy and electron balance of each reaction. You will also see the regulation of glycolysis by various factors, such as the concentration of glucose, ADP, NADH, and pyruvate, and the activity of hormones, such as insulin and glucagon. The most important regulatory step of glycolysis is the third reaction, where phosphofructokinase, PFK, converts fructose 6-phosphate into fructose 1,6-bisphosphate. PFK is inhibited by high levels of ADP and citrate, and stimulated by high levels of AMP and fructose 2,6-bisphosphate. Part 2. The Krebs cycle. If oxygen is available, pyruvate can enter the mitochondria and undergo further oxidation. The pyruvate is first converted into acetyl-CoA by pyruvate dehydrogenase, producing one molecule of NADH and one molecule of CO2. The acetyl-CoA then enters the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, where it is combined with oxaloacetate to form citrate. The citrate then goes through a series of reactions, generating three molecules of NADH, one molecule of FADH2, one molecule of GTP, and two molecules of CO2. The Krebs cycle completes the oxidation of glucose, releasing all the carbon atoms as CO2. In this part of the video, you will see the animation of the Krebs cycle, showing the structure of each molecule, the name and function of each enzyme, and the energy and electron balance of each reaction. You will also see the regulation of the Krebs cycle by the availability of substrates, such as acetyl-CoA and oxaloacetate, and the concentration of ADP, NADH, and FADH2. The most important regulatory steps of the Krebs cycle are the conversion of acetyl-CoA and oxaloacetate into citrate by citrate synthase, which is inhibited by high levels of ADP, NADH, and citrate. The conversion of isocitrate into alpha-ketoglutarate by isocitrate dehydrogenase, which is activated by high levels of ADP and NAD+, and inhibited by high levels of ADP and NADH. The conversion of alpha-ketoglutarate into succinyl-CoA by alpha-ketoglutarate dehydrogenase, which is activated by high levels of calcium and inhibited by high levels of NADH and succinyl-CoA. Part 3. Oxidative phosphorylation and the electron transport chain. The NADH and FADH2 produced by the Krebs cycle are then used to generate more ADP by oxidative phosphorylation. This process occurs in the inner membrane of the mitochondria, where a series of protein complexes, called the electron transport chain, transfer electrons from NADH and FADH2 to oxygen, forming water. The energy released by the electron transfer is used to pump protons across the membrane, creating a proton gradient. The protons then flow back through a protein called ADP synthase, which uses the proton motive force to synthesize ADP from ADP and phosphate. This process is called chemiosmosis. In this part of the video, you will see the animation of the oxidative phosphorylation and the electron transport chain, showing the structure and function of each protein complex, the movement of electrons and protons, and the synthesis of ADP. You will also see the regulation of oxidative phosphorylation by the availability of oxygen, NADH, FADH2, and ADP. The most important regulatory step of oxidative phosphorylation is the activity of ADP synthase, which is inhibited by high levels of ADP and activated by high levels of ADP. Part 4. Summary and conclusion. In summary, carbohydrate metabolism is the process of using carbohydrates as a source of energy for the cells. It involves three main pathways: glycolysis, the Krebs cycle, and oxidative phosphorylation. Glycolysis is the breakdown of glucose into pyruvate, which can be further oxidized in the presence of oxygen, or converted back into glucose in the absence of oxygen. The Krebs cycle and oxidative phosphorylation are the complete oxidation of pyruvate into CO2 and water, producing a large amount of ADP. Carbohydrate metabolism is regulated by various factors. I hope you get good.